Dear students, we continue topic sensory organs, and now we will talk about organ of hearing and balance. Organs of hearing and balance, both organs are secondary or sensoepithelial organs. They have very close anatomical and functional relationships. They have a lot of similarities in histological structure. That's why we will talk about them together. And we'll begin from organ of hearing or ear. So, ear or organ of hearing has three main anatomical parts. It's outer ear, middle ear, and inner ear. Outer ear includes ear pinna, external auditory canal, and tympanic membrane. Here we can see those parts. Middle ear includes tympanic cavity, hearing ossicles, and also Eustachian tube or auditory tube. And inner ear, it includes bony labyrinth and membranous labyrinth, which provide two functions. They provide hearing, and also they belong to organ of balance and equilibrium. So inner ear provides two functions, and it's part of two sensory organs, organ of hearing and organ of balance. And we'll begin from outer ear, and this outer ear, as you already know, it includes ear pinna, external auditory canal, and tympanic membrane. So ear pinna is covered with skin. Here we can see skin. Under the skin we can see fat, and basis of ear pinna it's elastic cartilage. Here we can see elastic cartilage, which is composed of elastic cartilaginous tissue, and it's covered with perichondrium. Its structure of ear pinna. Also, outer ear includes external auditory canal or external auditory meatus. It has cartilaginous part and bony part. Cartilaginous part has basis of elastic and particularly hyaline cartilage, and bony part has a basis of bony tissue, which is bony tissue of temporal bone. And cartilage with bone, they are covered with skin, which lines external auditory meatus. This skin has typical structure of skin, but it has some features, and the main feature is presence of special glands. They are ceruminous glands. They are modified sebaceous glands, and they provide production of serum. And also outer ear includes tympanic membrane. Here we can see tympanic membrane, and it has two sides. Outer side is covered with skin, which is continuation of skin of external auditory meatus, and inner surface is covered with mucosa of tympanic cavity. And middle part of tympanic membrane it contains a lot of collagen fibers and also elastic fibers, and they provide functions of tympanic membrane. From the middle ear we can see here it's malleus or hearing ossicle, which is attached to tympanic membrane. And tympanic membrane, it enhances sound waves and conducts them to the hearing ossicles, which are located in the following part of the ear, in the middle ear. So the main function of tympanic membrane is to enhance sound waves and conduct them to ossicles of middle ear. And next part of ear, it's middle ear. Here we can see it. It's tympanic cavity, three hearing ossicles, and also Eustachian tube or auditory tube. Here we can see this part of ear. So, hearing ossicles, they are composed of bony tissue, and they are very small. There are three ossicles. It's malleus, incus, and stapes. Here we can see that manubrium of malleus is attached to the tympanic membrane from inner side, and this part of ossicle, it receives sound waves. Next, it conducts them to head of malleus, from head of malleus to incus, and from incus to stapes. And those three ossicles, they conduct this sound wave, and also they enhance this sound wave, which is necessary for normal hearing. And here we can see stapes and bases of stapes. It closes oval window from inner ear. So basis of stapes conducts this sound wave to inner ear. Inner cavity, inner surface of tympanic cavity is covered with mucosa, which is covered with simple columnar epithelium or simple pseudostratified epithelium, as well as auditory tube, Eustachian tube, which is opened in the nasal cavity. Here we can see a tympanic membrane. Its malleus, which is attached to the tympanic membrane, its malleus head, and it conducts this sound wave to the following ossicles. And as a result of this conduction, sound wave goes from ossicles to inner ear. Cavity of middle ear of tympanic cavity is filled with air, but inner ear is filled with some special fluids. So now we are going to inner ear. Here we can see middle ear, 3 ossicles, and then they conduct this sound wave to the inner ear. Middle ear is filled with air as well as outer ear, and inner ear is filled with 2 fluids, endolymph and perilymph. And now I will talk about them. So it's inner ear, and as you already know, it provides 2 functions: function of hearing and function of sensation of balance and equilibrium. So it provides functions of 2 sensory organs. Inner ear is located in the pyramis of the temporal bones. Here we can see inner ear from 2 sides, and it includes 2 labyrinths, bony labyrinth and membranous labyrinth. Here we can see bone, and inside the bone we can see labyrinths. Outer labyrinth, it's bony labyrinth, it's bigger, and inside bony labyrinth we have the same but little bit smaller. It's called membranous labyrinth. So membranous labyrinth, it's located inside the bony labyrinth. Here we can see bony labyrinth, it's bigger. Here you can see bony labyrinth, and inside bony labyrinth it's membranous labyrinth. It's shown as blue structure inside violet structure. So it's membranous labyrinth and bony labyrinth. 2 same structures, structures with the same shape, but one is located inside another. So walls of bony labyrinth are composed of bony tissue of the temporal bone, and membranous labyrinth, its wall is composed mainly from connective tissue, and also there are some inclusions of epithelial tissue. And here we can see bony labyrinth inside the bone. Bone is brown here, and here we can see membranous labyrinth. It's included in the bony labyrinth. What are the features of bony and membranous labyrinth? They are filled with some fluids. Bony labyrinth, space between bone and membranous labyrinth, this space, it's white here, it's filled with special fluid called perilymph. Perilymph fills this space, and membranous labyrinth, inner labyrinth, this blue structure, it's filled with endolymph. Endolymph fills inner tubules of membranous labyrinth, and this space between membranous labyrinth and bony labyrinth is filled with perilymph, 2 fluids. And there are some anatomical parts of bony and membranous labyrinths. So first part, it's cochlea. Here we can see cochlea. Middle part, it's vestibule or vestibulum, and 3 semicircular canals. Here we can see them. 3 parts. Here we can see vestibule, middle part, cochlea, and 3 canals. So what are the functions? Cochlea, this part, only this part provides hearing. It contains sensory cells which react to sound. But we also have vestibule and 3 semicircular canals. They provide functions of balance and equilibrium. They are known as vestibular apparatus. Vestibule and vestibular apparatus. It provides a sensation of balance, location of our body in space, and also equilibrium. They also contain some sensory cells which react to the change of position of our body in the space, and also these parts, they react to the movement, vibration, our position of our head. And we will begin from this part, from cochlea, and first of all we'll discuss how sound irritates sensory cells. So let's begin from cochlea. Here we can see shape of cochlea, and we can see bony labyrinth of cochlea. Here we can see bony canals, and inside we can see canal, and basis of cochlea is called modulus. Here we can see modulus and canal which is rotated above the modulus. And here we can see cochlea, and we can see bony labyrinth and membranous labyrinth inside. Membranous labyrinth is shown in violet, and blue color it's bony labyrinth. This is filled with perilymph, and this thin canal is filled with endolymph. So here we can see modulus, bony labyrinth, and inside we can see membranous labyrinth. And here we can see modulus, bony labyrinth, and inside we can see membranous labyrinth. So, membranous labyrinth, here we can see, it's called scala media. It completely divides upper part and lower part of bony labyrinth. Here we can see bony labyrinth, and inside we have membranous labyrinth, and it divides bony labyrinth into two parts. Membranous labyrinth, it has triangular shape. It has apex, which is located near the modulus, near the axis of the cochlea, and outer part. And there are two membranes of membranous labyrinth which are attached to this part. So this part is called limbus, and there are two membranes, basilar membrane and Reissner's membrane or vestibular membrane. Two membranes, and they separate this triangle, which is called membranous labyrinth or scala media. Here we can see scala media, basilar membrane, and vestibular membrane. And outer side of this triangle, it's spiral ligament, and also is covered with stria vascularis of the inner ear. We'll discuss this later. So here we can see triangle, basilar membrane. Second membrane, it's vestibular membrane, and outer side of this triangle, it's stria vascularis and spiral ligament. Basilar membrane and vestibular membrane, they separate two additional ducts which belong to bony labyrinth. It's scala tympani and scala vestibuli, or vestibular duct and tympanic duct. Tympanic is lower and vestibular is upper, and between them we have endolymphatic duct or scala media, middle duct. And here you can see basilar membrane and cells, some special cells which are located on this membrane. They are known as organ of Corti or spiral organ. This organ contains sensory cells which react to sound. And let's look at histological slide, and let's find those membranes and ducts. So here you can see basilar membrane, vestibular membrane, and between them there is scala media, which is filled with endolymph. And also we have scala vestibuli, which is separated by vestibular membrane, and scala tympani, which is separated by basilar membrane. Both are filled with perilymph. Here we have perilymph, here we have endolymph. Lateral side of this triangle is spiral ligament, which is covered with stria vascularis. Here we can see stria vascularis. It's epithelial structure, epithelial tissue, and it's the only place of our body where epithelium contains blood vessels. Here we can see it. And those blood vessels, they produce endolymph. Here we have place where endolymph is produced. Basilar membrane, it has special epithelial covering, and those epithelium contains special sensory cells and another supportive cells, and they together are called organ of Corti or spiral organ, which is main organ which provides hearing. Those cells, they react to sound. What else do we have here? Here we have bony walls. It's spiral plate which begins from modulus. Here we have modulus, and spiral plate begins, bony plate begins from modulus. This spiral bony plate is covered with connective tissue, and connective tissue is covered with epithelium. And this connective tissue has name limb. It has two parts. It's basilar part and vestibular part of limbus or sides of limbus. Basilar membrane begins from limbus and goes to spiral ligament, and vestibular is continued into tectorial membrane. Here it's tectorial membrane, and it's vestibular part of limbus. And basilar membrane, it has special cells of organ of Corti. And also we have Reissner's membrane. It's much thinner than vestibular membrane, than basilar membrane, and it hasn't sensory cells. It's covered with simple squamous epithelium, as well as walls of vestibular duct and tympanic duct, or scala vestibuli and scala tympani. And also we have here spiral ganglion, nervous cells, bipolar nervous cells, which dendrites are going to sensory cells. So sensory cells or sensoepithelial cells are present here, and they send information to dendrites of bipolar cells, and dendrites are going to the perikaryones of neurons, which are located near the modulus, and they form spiral ganglion. It's located here. Here we can see another side from cochlea. It's vestibular duct, basilar tympanic duct, and scala media, or scala vestibuli, scala tympani, and scala media. It's basilar membrane and it's vestibular membrane, organ of Corti, and tectorial membrane. Beginning of tectorial membrane and basilar membrane, it's called limbus. Here we can see stria vascularis of the inner ear, and its lateral side of scala media. It's only place in our body where epithelium contains blood vessels. Here we can see capillaries, which are located between epithelial cells. It's place where endolymph is produced. And let's look at organ of Corti. Here we can see organ of Corti. It's located on the basilar membrane, and it has two groups of sensory cells. Inner cells, inner sensory cells, they form one row usually, and outer sensory cells or sensoepithelial cells, they form usually 3 rows. Here we can see 3 rows of cells, and inner cells. They are called hair cells because they have processes on their apical pole which are called stereocilia. They have very important role in the sensation of sound. So here we have hair cells, inner cells, they form 1 row, and outer cells, they form 3 rows. And also we have supportive cells or maintaining cells. They maintain hair cells. They are located under them. Here we can see hair cells. Outer, they form 3 rows, and inner 1 row. And also they have maintaining or supportive cells which maintain hair cells. And here we can see hair cells, outer and inner, and maintaining cells. Also we have columnar cells or cells of columns, and they separate tunnel of the organ of Corti. It's triangle opening between inner and outer hair cells. And here we can see dendrites of sensory cells which are coming here to hair cells, and they receive information from hair cells about sound and bring this information to spiral ganglion. Here we can see hair cells. They have stereocilia, hairs on their apical surface, and they are irritated by special tectorial membrane. Here we can see tectorial membrane which begins from limbus, and it covers stereocilia of hair cells, and it touches them. And when hair cells are moving, this tectorial membrane touches them, and it causes irritation of hair cells. And later we'll see how they are irritated. So main in the hearing, it's when tectorial membrane irritates stereocilia of hair cells, and they generate impulse which is uptaken by dendrites of sensory cells, and dendrites from sensory cells, they bring this information to spiral ganglion. Here we can see stereocilia of hair cells. We can see 3 rows of stereocilia, and we can conclude that there are outer hair cells. Inner hair cells, they form 1 row. So here we can see a hair cell with stereocilia, and when stereocilia are moving, for example, when tectorial membrane touches stereocilia, a cell is activated, it's excited, and it generates impulse which is uptaken by dendrite of sensory cell, and this impulse goes to sensory cell in spiral ganglion. So organ of hearing, it's a secondary organ or sensoepithelial organ because it has sensoepithelial cells. And here we can see appearance of hair cells. There are inner cells with stereocilia, they form 1 row, and outer they form 3 rows. So how stereocilia will be irritated? Hair cells, together with supportive cells, maintaining cells, they are located on the basilar membrane. When basilar membrane is moving, when sound wave is going through the cochlea, through the perilymph of cochlea, it moves sound, moves basilar membrane. Basilar membrane is moving, and it moves hair cells which are located on the basilar membrane. And hair cells, they touch tectorial membrane. So tectorial membrane isn't moving. Hair cells are moved by basilar membrane, which is moved by sound wave. So sound wave moves basis, it moves the floor of the hair cells, and hair cells, they touch tectorial membrane, and tectorial membrane irritates their stereocilia. So main in the hearing, it's to move basilar membrane. Basilar membrane moves, and hair cells touch tectorial membrane, and stereocilia are activated, are irritated, and hair cells generate nerve impulse. So how a basilar membrane is moved, how this membrane becomes to be movable. We have 2 scala vestibuli and scala tympani. They are filled with perilymph. This scala media is filled with endolymph. So these fluids, they are moved by sound wave in scala vestibuli and scala tympani. Here we can see cochlea, and upper scala, it's scala vestibuli, and lower, it's scala tympani. They have connection on the apex of cochlea. It's called helicotrema or helicotrem. It's opening between 2 ducts. Both are filled with perilymph. It's bony labyrinth. So here we can see basis of stapes, and it closes oval window. Here we can see oval window. It's opening of scala vestibuli. Scala tympani also has opening. It's round window, and it's closed with secondary tympanic membrane. But no bones are present here. Only oval window of scala vestibuli is closed with bases of stapes. So when sound comes here, it moves tympanic membrane. Tympanic membrane moves malleus, it moves incus, incus moves stapes. Basis of stapes is located in the opening of oval window, and it conducts this sound wave to perilymph of scala vestibuli. And this wave runs through scala vestibuli, goes to the apex of the cochlea. Here, via helicotrema, it goes to tympanic duct to scala tympani and runs in the opposite direction and goes to the opening of the tympanic duct to the opening of scala tympani and moves secondary tympanic membrane in the oval window. And when sound wave runs here, it moves basilar membrane. Here we have vestibular membrane or Reissner's membrane, and here we have basilar membrane. And when sound wave runs via scala tympani, this sound wave moves basilar membrane. This membrane is moved. First of all, sound wave goes to the apex of cochlea via this scala vestibuli, and in opposite direction it runs via scala tympani and moves this membrane. This membrane moves, and it moves hair cells. Hair cells touch dictorial membrane, and they are irritated, and it results in the generation of nerve impulse, which goes to spiral ganglion. So here we can see that sound goes to the apex of cochlea. Here we can see to apex of cochlea, and it goes in the opposite direction, moves basilar membrane, and this sound wave excites from cochlea via round window, which is closed with secondary tympanic membrane. And also, you know that there are high sounds and low sounds. Why some sounds are high, why some sounds are low, and how do we distinguish them? Basilar membrane has collagen fibers. Here we can see collagen fibers, and their length is different in different parts of basilar membrane. In the basal part, collagen fibers are short, and in the apex they are long. And they remind us different musical instruments, and short fibers are irritated by high sounds, and long collagen fibers near the apex of cochlea, they are irritated with low sounds. And middle length collagen fibers, they are irritated by middle sounds. So when we hear high sounds, collagen fibers in the basis of cochlea are moved, and they irritate corresponding hair cells. So basis of cochlea provides hearing of high sounds, and apex of cochlea, it provides hearing of low sounds, and middle part of cochlea provides hearing of middle sounds. So here we can see base and apex, short collagen fibers and long collagen fibers, and we can see when we hear low sounds, they move long fibers like musical instruments, and we hear them near the apex. And opposite situation, when we hear high sounds, they irritate short fibers, and they are moved near the base of cochlea, and we hear high sounds. So apex hears low sounds, base hears high sounds. And here we can see stapes conducts sound wave via oval window to scala vestibuli. Sound goes to the apex of cochlea, turns back and goes through the scala tympani, moves basilar membrane. Basilar membrane is irritated and hair cells are activated corresponding to some degree of sound, and it results in the generation of nerve impulse. Here we can see direction of sound. Sound runs via upper scala vestibuli, goes to apex of cochlea and turns back via helicotrema and runs in the opposite direction, and on this way it moves basilar membrane and it irritates sensory epithelial cells of organ of Corti. And now we are going to organ of equilibrium or organ of balance. As you already know, it's very related with cochlea, which provides hearing, and its inner ear, it has cochlea, it has vestibule, and 3 semicircular canals, which provide balance and equilibrium. So, organ of balance and equilibrium is located in the inner ear, which is located in the pyramis of the temporal bone. It also has bony labyrinth and membranous labyrinth, which is located inside the bony labyrinth. Here we can see membranous labyrinth, which is located inside the bony labyrinth, and there are some sensory regions. Here we can see organ of Corti or spiral organ inside the cochlea. It provides hearing, but also we have 5 additional sensory zones. Here we have 2 sensory zones in the vestibule, and also we have 3 semicircular canals, which also have sensory zones. So what are the special features of organ of balance? We have vestibule and we have 3 semicircular canals. Vestibule in bony labyrinth is common cavity, but in the membranous labyrinth it's divided into 2 parts. It's sacculus and utriculus. Sacculus and utriculus, and also we have 3 semicircular canals. They have ampulla, and there are cristae of ampulla of semicircular canals. So sacculus and utriculus, they provide sensation of movement, linear movement, when we are moving up and down, right and left, forward and back. We irritate these sensory cells in the utriculus and sacculus. And ampulla of semicircular canals, those sensory cells, they react to movement of our head, to rotation of our head, and they react to this kind of movement. And also sacculus and utriculus, they react to the vibration and gravitation. We can feel where is basis, where is ground, even if we will close our eyes. So utriculus and sacculus, they are responsible for this. So sacculus and utriculus, they have special sensory regions which are called otolith apparatus. There is usual epithelium which lines membranous labyrinth, but in some places there is special sensory epithelium which forms otolith apparatus. There are supportive cells, and also there are sensory hair cells. We can see them here. They are very similar to hair cells of the inner ear of the Corti apparatus. Here we can see them. But they haven't tectorial membrane. They are covered with special gelatinous material which contains small stones which are called statoconia, and together gelatinous material with statoconia, it called otolith or otolith apparatus. When we are moving, when we move our head, those stones also are moved because of gravitation. And this gelatinous material, this jelly, these stones, they irritate stereocilia of hair cells, and cells are excited. They form nerve impulse, which is uptaken by dendrites of sensory cells. So in this otolith apparatus we haven't tectorial membrane, we have special stones in jelly which irritate hairs of the hair cells. And we feel our movement, and we feel where is ground, or we feel vibration. And here you can see histological slides of otolith apparatus. There are hair cells and jelly which covers those otolith apparatus. And here we can see hair cells, their stereocilia and one long kinocilia. They are located in jelly which contains otoliths. So hairs, stereocilia and one long kinocilia, they are moved by jelly with small stones. Cell is excited, it forms impulse, it sends information to dendrite of nerve cell, and this information goes to central nervous system. And here we can see how stereocilia are moved during movement of our head. When we are moving, otoliths, under influence of gravitation, they also are moved, and they move jelly, which moves stereocilia of hair cells. Also, we have semicircular canals. We have 3 canals in each ear, and they have ampulla, and ampulla they have cristae. Cristae are covered with special sensory cells, hair cells, as well as in the otolith apparatus, but they haven't otoliths. They are covered with special cupula, jelly cupula, which covers those otoliths. Here we can see cupula, cells with stereocilia, and also there are supportive cells. And these semicircular canals, they react to rotation of our head. So when we are rotated, cupula moves together with fluid, with endolymph, and it irritates stereocilia of hair cells, and we feel rotation. Here we can see cristae of semicircular canals. It's ampulla, and it's cristae, it's cupula, and it's otolith apparatus, its neighboring structure. So this type of sensory cells, they react to rotation of our head. So we have discussed all sensory organs. That's all, and thank you for your attention.